All right. So we're talking about meiosis, and I said, well, meiosis, to produce gametes, haploid cells, it's a two-step process. It's meiosis 1, which is reduction division. That means we're going to take our 2n that's in the parentheses and split it. So we're going to have this 2n kind of situation, right? And within there, with the other end separated out now, okay? And that's what reduction division is we're separating the chromosomes to make haploids. But we keep the copies together, so in meiosis 2, we must separation of chromosomal copies, or chromatids. That's why meiosis 2 is similar to meiosis 2 is similar to mitosis. Okay? Because it's separation of copies. Okay? The difference is this is separation of diploid copies, right? It's going to produce 2 diploid cells. Because these are diploid. These sister chromatids that make daughter cells, these are diploid daughter cells. They're diploid daughter cells, guys. They're 2n. We make 2n. We make two cells, 2 daughter cells from 1. In ladies, that's not the case. From 1, you make 1. In guys, from 1, we make 4. Huh? Everyone agree? All right? Because we covered that. All right? We covered that. And I'll go over it again with you guys. So, all right. So then what winds up happening is, so we said, all right, well, so the whole idea is Mendel helped us to understand about this idea of genetics versus the physical trait. He looked at pea plants, not humans. But what he saw in pea plants, actually, and the data he collected when he did these pea plant experiments, because he had nothing else to do with his life. All right? So this guy actually, he would literally parcel out land across the monastery, right? That was isolated. So he could have his true breeds here. Hey, mom, dad, your progeny, I'm just going to keep, because again, they're pea plants, right? They're not humans. We're just going to have you keep reproducing amongst yourselves. So you're all going to be inbred. So it's all going to be pure. Got me? So he did that with red, he did it with white. Boom. 2 different areas of the monastery. Of course, he had a shit ton of area in the monastery is huge. Right? So, and then it dawned on him, hey, what if I take a red flower, a male red flower, and a female white flower, and I introduce them, and what comes of that, I plant it in a different area of the monastery and allow it to grow. And sure enough, what type of flowers do you think it produced? Pink colored flowers. So the color of the flower of that generation, of the mix of the 2 purebreds, produced a heterozygote that was neither like one or the other. Well, that's the perfect example of the codominant trait. Well, the ABO blood system is the perfect example in humans of a codominant trait, right? And that you get A and B, you express A and B together. Okay? That's how it is. So again, we go back to blood. All right? As we finish up this semester, you see that there was a method to my madness, especially if you have me for 2085. All right? Because I kind of repeat myself regularly. All right? So genetically, it's associated with our genotype. And physically, well, that's our phenotype. And so I said, well, that's true because we get protein from DNA. We got to go DNA to protein, but to do that, we got to do RNA. So then I went through and did a review of DNA, RNA, protein, and then to go from DNA to RNA, you got to do transcription. From RNA to protein, you got to do translation. And then for DNA to make new DNA, I got to do replication. That occurs in the S phase of interphase, and then I wrote the cell cycle. And the cell cycle has G0, which is cell senescence, when the cell is not involved in the cell cycle. Because cells aren't always involved in the cell cycle. All right? They're told when to go in the cell cycle. And when they do go in the cell cycle, right? When they do go into cell cycle, then they'll go through G1, S, G2, and either mitosis or meiosis, depending on the signals, guys. And if I'm in the gonads, then if I'm in the gonads and we're talking about testicles and we're talking about sperm development, by the way, guys, we do not start producing sperm until we hit puberty. So up until then, this is why our levels of testosterone, this is why, again, male and female children have same body habitus. What do I mean by that? Toddlers, children, babies, children, right? They have the same body habits. You can't tell male from female unless they drop their drawers. You see some kids with long hair, super cute, man, you think they're a girl. Especially if like mothers let them go a little haywire, yeah. Right? I'm just saying, right? If mom's like, yeah, whatever, you know, kid starts putting dresses on, so we're gonna confuse him for a girl? Hey, he wants to wear a tunic, somebody's gonna think he's a girl, he's got long hair. No, don't get upset, right? You putting them on in these, you know, neutral colors. Okay, I understand, right? But somebody's gonna get it wrong. So male, female children have same body habits, guys. Why? Because until you hit puberty, guess what? You don't need none of these reproductive structures. You don't need none of them. But when you hit puberty, then ideally, that's when you're supposed to reproduce. Right? At least that was the idea back in the day. So girls were getting married off at 13, right? To guys that were 15. And then you guys are having, you know, and then sure enough, the body has the ability to recuperate quicker. I can't lie to you. But what happens is every child after that subsequently will cause the body to weaken. And if the female doesn't take care of herself, then sure enough she'll probably wind up giving birth to a child and wind up dying, which is usually the case back in the day. All right, if the child didn't die with the mother, that is one of the reasons why we created the National Institutes of Health, try to reduce the number of the fetal mortality in the United States. You understand? If we were looking at the reasons why fetuses were dying, or why newborn babies were dying. All right? The population of people pay taxes, don't you? Otherwise, you got no federal government. Sure out, right? So male and female children have the same body habits. So that until we hit puberty, who's making the hormones that we need? That kind of keep the gonads properly aligned and doing everything else like the secondary sex characteristics. By the way, what are secondary sex characteristics? Pubic hair. You know what they call pubic hair? Well, we call them Tanner stages of development, guys. Tanner stages of development. You can look them up. 4 stages. 4 stages is the last stage. First stage is the first stage. Your stage 1, Tanner stage 1, you should just showing like sparse pubic hair, very fine, down in the area. Right? Little kid won't have any hair there, boy or girl. Then they'll start to show fine hair. Then the hair will start changing texture and it'll start to populate. They'll start to grow. The bodies will start to grow. Women will start developing. The breast tissue will start to develop. Okay, so that means what? That means fat cells, connective tissue cells, yes? And a lot of that's there and lying dormant. And then comes online for development purposes. And that's separate of, again, when you're pregnant, right? And then of course once you give birth, because once you give birth, then your hypothalamus is going to release prolactin. Your prolactin release will cause the production of milk, right? And then separately oxytocin released by the hypothalamus to the posterior pituitary gland, right? Would cause the smooth muscle contraction, which would allow the ejection of milk. So you need lactation, which is driven by prolactin. So prolactin is for lactation, and oxytocin is utilized for milk ejection and delivery, what we call parturition. Okay? Delivery. So now we're talking OBGYN. All right? And so, again, because we're talking about female and male reproduction. All right? Now, so what Mendel discovered was, he discovered, one, that there was codominant traits, that there were codominant, and that he didn't know quite how to explain that. He didn't know how quite to call it. But what he was known for more so than anything else, because he did develop this data, this initial data, he just didn't know how to explain it. What he did look at is he looked at the color of the pea, and he looked at the shape of the pea. He also looked at the stalk, he looked at the flower, he looked at all these different physical characteristics. And then again, for each of the pure plants, and then as he mixed them, he then reclassified what he was seeing in the physical traits of the crosses. Do you get me? And so it's interesting that separately he develops an idea of there being a dominant and a recessive type system. Meaning, so okay, follow me for a moment. In a dominant type system, you only need one gene to be bad. Just one or two genes. You got me? So to have disease, 1 out of 2. 1 of 2 genes bad. Okay? 1 of 2 genes bad for a dominant disease. Okay? Or you could say for a dominant system, say protein system, you need one particular gene to be dominant over the other one. You only need one gene. You follow? To be dominant over the other one. Then in a recessive, for recessive diseases, so for dominant diseases, you need 1 of 2 genes to be bad. For recessive diseases, you still need one of, in this case, you'll need 2 of 2 genes to be bad. Sorry. You'll need both genes to be bad that you inherit to develop the disease. Okay? Otherwise, you're a carrier. So you're a heterozygote. Now watch. Very basic. Big R, little r. So if I'm purebred versus purebred of the opposite, and I cross, every generation I get is a carrier, but heterozygotes. Sure enough, when I take those heterozygotes from this generation and I mix them again with each other, then I get this strange pattern. And you get, so if I get a heterozygote cross with a heterozygote, and this is on the homework, you're going to get a 1-to-1 ratio of heterozygote, homozygotes dominant to heterozygotes to homozygotes recessive. But it's a 3-to-1 ratio for physical trait disease, or whatever, for physical trait, or phenotype, physical ratio. All right. So look that stuff over. It's in there, right? That's the inheritance stuff. Right? And yeah, pregnancy. You're not allowed to questions though. More so about like implantation and delivery, the development of the fetus. So, not too much about the development of the fetus though. I'm not going to kill you guys with embryology. I just want you to know the basic stuff.
Thanks, Professor.
Yes.
Have a
good day.
You too.
All right? Yeah.
Bye, thank you.
Bye. Have a good day. Oh, yeah. What did I say? I said co-dominance. I meant incomplete dominance. Yes. Yeah, because neither one of the two are dominant. And so they're both, it's sort of like a co-dominant type system, because like what's really happening is the both colors are being expressed at the same time, but neither one of them is being expressed in their entirety, and that's the difference between the incomplete dominance versus the co-dominance versus dominance versus recessive. Yeah, that's right. Both are expressed fully. Yeah, so you're correct. Co-dominance versus incomplete dominance. And this is all kind of, and this is, and again, this is Mendelian genetics kind of expanded on. Not even Mendel could explain the incomplete dominance or even co-dominance. He couldn't explain. That didn't come till later, but he had the data. He just kind of put it up the side. He was like, yeah, I can't really do it. I don't even know what to call it. And they didn't know what to call it. They didn't even know what to call the gene.
I think he was lucky with his choice of P too, because there are a whole bunch of
yeah, he was lucky.
It was linked.
And that, not only that he was careful as to how he collected the data. He was very precise, meticulous, and the fact that they found it later and showed, and it was applicable to some of the systems that they were looking at, where they understood complete dominance and incomplete dominance and co-dominance, which I forgot completely about incomplete dominance. Yeah. But you're correct. So the dominant type diseases or disorders are going to be driven by, if you get one or two genes bad, then you got the disease. It just is what it is, and that means that if you get this genotype,
it's like achondroplasia,
then you're phenotype. I'm going to give you a couple diseases. I'll give you guys a couple diseases.
I've been writing down the ones that you know as clinicals. I'm hoping.
Yeah. So don't forget the one that we talked about, the pseudohermaphrodite with the SRY gene.
Yeah.
Because that's a sex linked trait, okay? And so the development of male genitalia is sex linked. It's linked to the Y chromosome. And that's
so once you have your Y, you're a male.
That correct. If
any format, whether it's XXY or
if you have, that's right. You still, that's right. So you have the SR1 gene. Yeah. So if you get the XXY, that's a little tough, because what winds up happening is which one do you?
Nice change.
Yeah,
they say this, I think one time it says once you have the Y, you have the maleness, meaning you're male. But
genetically, you're definitely male. Yeah,
sometimes you have syndrome.
Yeah, if you have XXY, where you get this non-disjunctional event, which we'll talk about on Tuesday, I'll draw this meiosis again the way I did, but I'm going to explain to you more as to what's happening in meiosis, because this is what's really important here. It's the cycle of meiosis, and it's that synaptic complex and that crossing over event that allows for the exchange of genetic material, and that's what gives diversity to each of the chromatids. Because each chromatid, when that happens, will be different. Because one will be untouched, and the other two will be altered, and the other won't be untouched. And so when they all get split apart to make sperm, then you got four sperm who each of the chromosomes could be altered in that way. And that's, again, this happens independent. The crossing over event, just like the independent assortment of each of the chromosomes, the selection and pulling from each one to each pole is independent of the other. The crossing over events are also independent of each other, which is really, really interesting, because that means it could happen or not happen. And then this also gives play to